Welcome back to this next video in which we are discussing HPVs and cervical cancer. Okay, so let's finish now the HPV life cycle. So we have finished the early portion of the HPV life cycle, which is the bit that we're going to discuss in most detail, and we've only got the late portion of the HPV life cycle to now go over. So let's just summarise where we've got to. Overall, the result of what has happened so far is that we have loads of mid-zone cells that have nuclei absolutely stuffed full of viral genomes. We have loads of viral genomes at this point. Obviously, we now need to package them up into capsids to produce the finished viral particle. So, this now is the late portion of the viral life cycle. So we'll come up here and we'll study the late portion of the viral life cycle. So what happens now is something called the early to late shift, or the early to late switch, which is a switch in which viral proteins are actually going to be being produced. So we're going to go from making the early proteins to now making the late proteins. So this is going to be called the early to late shift. And it's not very well understood what actually triggers this, how it's all coordinated, but it's a marked shift in which proteins are being produced. So remember, in these mid-zone cells, in the early portion of the life cycle, we were producing all of these early proteins, E1, E2, E4, E5, E6 and E7. We've discussed the role of E6 and E7 in making the cells proliferate really fast and therefore triggering viral genome amplification. We know that E1 and E2 are absolutely essential for genome replication. E4 and E5 don't have very well understood functions, so we haven't discussed those. These proteins were expressed in the early portion of the life cycle. They are going to be we're going to stop the production of these proteins now. We're going through the early to late shift. So these stop being produced now, and their levels will gradually fall as they're gradually destroyed in the cell by the proteasome and other proteostasis mechanisms. So their levels will start to fall, and we're now going to go over to producing the late proteins. But before I talk about the production of the late proteins, let's talk more about the consequences of the early proteins going down. So in these viral stuffed cells, so this mid-zone cell here that's stuffed full of viral genomes, and it could have thousands of viral genomes in its nucleus now. It now is dropping its expression of all of the early proteins. So these proteins are all going down in the cytoplasm of the cell. What's that going to mean? It's going to mean that the cell is going to stop dividing at the ridiculously rapid rate, because E6 and E7 were the ones driving that very rapid proliferation. Now they've gone down, it will stop proliferating at that horrendously rapid rate. And that means that it can continue on with the differentiation process. Remember, if a cell is dividing, it cannot differentiate. If a cell is differentiating, it's not dividing. The two are, they can't go together. So when these mid-zone cells were dividing at a fantastic rate because of the E7 protein, they weren't going through their normal differentiation process that is part of the normal physiology of the squamous stratified epithelium. Now that the early protein expression has gone down, they can continue on with their differentiation process. So let's show this here. So this cell will now continue on with the differentiation process, and let's say it's now moving up into the superficial zone here. So I'll draw it now as a nice squamous flat cell, and it hasn't yet lost its nucleus, or we're dealing with a non-keratinized squamous stratified epithelium where it wouldn't lose its nucleus anyway. So I've still got the nucleus here, and if we're dealing with a keratinized squamous stratified epithelium, this is supposed to be a low down cell in the superficial zone, so it hasn't yet lost its nucleus. So this layer now is meant to represent the superficial zone of the squamous stratified epithelium. And that arrow that we've got there, of course, is the differentiation process that is now allowed to continue because the expression of the early proteins has gone down. Okay, so when early proteins go down, differentiation occurs, and of course, this differentiating cell will still have all of the viral genomes in its nucleus, so those won't have suddenly gone away, they're still here. So here they are. Right, So what now happens is the late proteins are going to be expressed now that we're up in the superficial zone. So L1 and L2 are going to be expressed. So let me draw these on. So we'll have L1 here in blue, so L1 starts to be produced, and then we'll have L2 as some bright yellow colour here. So here is L2. So remember, these are the major capsid protein and the minor capsid protein, and I've just drawn one of each of these. In reality, you're going to be producing a huge number of them. Loads of L1 proteins and loads of L2 proteins are going to be produced. Obviously, far more L1 proteins than L2 proteins. And this again will be using host cell protein synthesis machinery, because remember, the viral genome doesn't code any of its own protein synthesis machinery, so we're completely reliant on the host cell to produce those proteins. Okay, so once we're up in the superficial zone, you start producing the late proteins L1 and L2, and then as the cell continues to move up in the superficial zone to the more superficial layers, what will happen is the viral genomes will start moving out of the nucleus and into the cytoplasm. And of course, if we're talking about a keratinized squamous stratified epithelium, the nucleus will start to go anyway. But even if we're talking about a non-keratinized squamous stratified epithelium, they still move out of the nucleus into the cytoplasm, and then you'll start getting the viral particles being formed. So here we go. It's now moving up to the more superficial parts of the superficial zone, and what we'll start to get is viral particle production. So here are some capsids being formed out of the L1 and L2 proteins. And let's imagine, as far as this picture is concerned, that we're talking about a keratinized squamous stratified epithelium, so it's lost its nucleus just to make more space for me drawing virus particles. Those are the capsids, and here are the genomes inside of the capsids in green here. Okay, so up here, as the differentiation process continues, you get viral assembly occurring. And then the final part of the life cycle is that this superficial cell will be shed, and of course when it's shed, it will be absolutely stuffed full of the new HPV virion particles, then the shed cell will be mechanically injured at some point, because it's being shed into the environment. Something will injure it. It will gradually, lose its integrity, and all the virus particles will be released. So understand that important point, that the virus particles do not get out of the cell until the cell has been detached from the human body and is off. And then the cell will break open and the virus particles will be released, and you'll have produced a whole bunch of new viral particles. So this portion of the life cycle that we've just been through, all of this is what's called the late phase of the viral life cycle. And that is the viral life cycle of HPV complete. What I want to do now is summarize the life cycle and then draw your attention to a few interesting points about the life cycle. Then we'll talk about the thing that I promised to talk about right at the beginning, which is transmission. Okay, so let's summarize the life cycle again then. So, human papillomaviruses, all of the different types of them, they infect squamous stratified epithelia. We know that there are two major divisions of human papillomaviruses, those that affect cutaneous epithelia and those which affect the non-keratinized mucosal epithelia. All of the human papillomaviruses, however, start by infecting the basal cell. And this is actually an interesting point about human papillomaviruses. You'll notice that their life cycle involves loads of different cells. That's unusual for a virus. Usually a virus infects one cell, uses that cell to make loads of the new viruses, and then the viruses leave that cell. It doesn't usually involve multiple different cells at multiple different stages of an epithelium like this. Human papillomavirus, its life cycle is completely entangled with the differentiation process in the squamous stratified epithelium. It's completely entangled and in flow with the life cycle of the cells in a squamous stratified epithelium. So that's a first interesting point that I wanted to draw your attention to, that it's an unusual viral life cycle. So, the human papillomavirus infects the basal cell, and remember, to get access to the basal cell, you either need to have a microabrasion of the squamous stratified epithelium that allows it to get to the basal cell, or you need to have incomplete metaplasia, and that is why the cervix is so vulnerable to HPV infection, because you will have incomplete metaplasia for a very, very long time on the squamocolumnar junction of the cervix, because the metaplasia process takes decades and decades and is ongoing, and at the squamocolumnar junction you will have incomplete metaplasia. So that's why the cervix is so vulnerable to HPV infection. So we don't know much about the entry process, so we summarised it as an arrow. The genome gets into the nucleus of the basal cell. It doesn't code any of its own RNA polymerase machinery or DNA polymerase machinery, so it's therefore completely dependent on host cell machinery, and the first thing it does is express the two early proteins E1 and E2, which are essential for it being able to copy its genome. These bind to its origin of replication, and they're involved in recruiting the host cell DNA polymerase machinery onto the origin of replication, so that it can then replicate the viral genome. What will then happen is the basal cell will divide as part of normal physiology to produce another basal cell and a daughter cell that moves up. In that division process, the viral genome will be copied more times than the host cell genome, but still not that many times. You'll get low copy numbers of the episomes in both the basal cell and the daughter cell that's moved up. Just to emphasise that this basal cell still has viral genomes in, and when it divides again to produce another daughter cell and another basal cell, that new daughter cell again will have viral genomes in. So you're going to continuously be producing more daughter cells that have viral genomes in. So this is a source of more viral genomes, as well as just what we're about to talk up here. So, continuing now the story for the daughter cell that moves up into the mid-zone, yes, we have some viral genomes in here, and yes, even if we didn't have this high productive phase of the life cycle, we would still be producing more virus particles, but we can hugely exaggerate this effect by having another amplification phase, and this is the major production phase of the viral life cycle. So what now happens is, in this slightly differentiated cell that's moved into the mid-zone, the virus expression activates. The virus starts to express E1, E2 at a higher level than before, and also the other early proteins E4, E5, E6, and E7. We don't understand fully why this happens, but E4, E5, E6, and E7 are only expressed once it's undergone a bit of differentiation. They're not expressed in the basal cell layer, only in the mid-zone layers. E4 and E5 we drew a great big cross across, we don't know much about those. E6 and E7 we saw are incredibly famous, incredibly powerful proteins. E7 causes the cell to rapidly divide by activating S-phase gene expression and therefore forcing the cell to divide. It's more powerful in the high-risk human papillomaviruses than the low-risk human papillomaviruses. E6 in the high-risk human papillomaviruses with very powerful E7 proteins inhibits the p53 pathway, and which is really important because E7's aberrant cell division would activate the p53 pathway if you didn't do something to stop that activation. So E6 is the partner in crime with E7, and overall they result in these mid-zone cells dividing extremely rapidly, which then can produce a wart in some cases, and also the viral genome will be amplified a huge number of times. So you end up with loads of cells in this mid-zone, just like this picture that I've got here, where they have nuclei stuffed full of viral genomes. And I've said it before, but I'll say it again, because it's very important. You get a huge number of viral genomes in here. It can go up to thousands. So we call this a high copy number of episomes there. And that's the end of the early phase of the life cycle. You then go into the late phase of the life cycle, where now the early proteins, their levels in the cell fall, and this allows the cell to stop dividing and commence the differentiation process once again. So it moves up into the superficial zone, where you start expressing the late proteins L1 and L2, the major and minor capsid proteins. And then as differentiation continues, the viral genomes move out of the nucleus into the cytoplasm, and you package up new viral particles. The cell is then shed from the surface of the squamous stratified epithelium, and then it will break open and the human papillomaviruses will be released. And that is the viral life cycle. That's how we produce more human papillomaviruses. Okay, so now let's talk about transmission, the thing that I promised to talk about at the start. So I'll just put it as a little title here. Transmission. So transmission is by skin-to-skin contact if we're talking about cutaneous HPVs, and I think it's best to start with cutaneous HPVs. So if you've got cutaneous HPV infection, let's say on your hand, and then you touch someone else's skin where the cutaneous HPV infection is, that can then lead to them getting an HPV infection. The reason is that where you have an HPV infection, you are shedding cells that are full of HPV virus particles. So if you then touch the skin of someone else, you're covering them in human papillomavirus particles. And then if they've got a microabrasion on their skin at that point that you've touched them, the human papillomaviruses can get access to the basal cells, and then that's how you transmit the cutaneous HPV infection. Talking now about, so skin to skin is for the cutaneous HPVs. Talking now about the mucosal HPVs infections, these are generally transmitted by sexual contact. So for instance, let's start off with imagining that we have HPV infection of the oral cavity mucosa. If two people were kissing and one of them had a mucosal HPV infection of the oral cavity squamous stratified epithelium, then you can imagine that that will transmit the HPV virion particles into the mouth of the other person, and then they are at risk of getting an HPV infection there as well. Alternatively, coming down to the external genitalia, you can imagine how if a boy has a HPV infection of the preputial mucosa, and remember this might not be visible, it might be visible and you might have a genital wart there, but it might not be visible as well. You can imagine how this could then be transmitted to the vulva mucosa of a female, or the vaginal mucosa of a female, or indeed the cervical mucosa of a female. I'm not going to spell out these stories. I'm sure you've got imaginations enough to be able to come up with the explanations yourself. And you can also imagine how it could be transmitted to the anal mucosa. So the one thing that I do want to say is the fact that condoms are not particularly effective at stopping HPV transmission but through sexual contact. And the reason is that, unlike with HIV, where if you use a condom, the transmission is nil, it completely cuts off transmission. It's not the case for HPV infections, and the reason is that the virus is actually on the mucosa rather than in the semen or the vaginal secretions. So even if let's say the boy has a preputial mucosa HPV infection and he puts on a condom, when he's putting on the condom he will most likely get the HPV virus particles all over his hands, and unless he then goes and sterilizes his hands before he then touches the girl, you can imagine how the HPV infection can then still spread to the female regions. So that's why condoms are not particularly effective at preventing the transmission of HPV infections of genital warts, whereas they are, of course, incredibly effective at preventing the transmission of viruses such as HIV. Okay, so that's all I want to say about transmission. The final thing that I'd like to go over in this video, and I think I'll put it in this little space here, is the immune response to HPVs. Now, I'm not going to say much about this because not much is actually known about the normal immune response to an HPV infection. So, immune response. We'll talk a lot more later about the immune response triggered by vaccination, but the immune response triggered by vaccination is very different to the immune response that you get if you just have a HPV infection on one of your squamous stratified epithelia. So, Immune response is not terribly effective at clearing HPV infections. It takes an incredibly long time, and you'll know this just from common knowledge. Warts can last for years and years and years, so it can take the body an incredibly long time to actually launch an immune response that is capable of destroying the HPV infection. And the reason that it takes so long, and I will just put this as a big point, it's extremely slow. The reason it takes so long is that the HPV infection is not actually acutely deadly at all. I know the whole video is about how bad HPV infections are of the cervix and how it can lead to cancer, but that's a process that takes decades. These infections are not acutely deadly. When you compare them to an infection like malaria or some horrendous septicemia, bacterial septicemia, they are not acutely deadly at all. They are really quite harmless, and that's the reason that you don't launch a particularly powerful immune response against them. So some statistics now. We reckon that about 90% of HPV infections will be cleared in 2 years. So 90% of HPV infections cleared in 2 years. The other 10% will not. And that other 10% we call persistent HPV infections. And we're going to see that it is persistent HPV infections by high-risk human papillomaviruses in the cervical squamous stratified epithelium that then leads to cervical cancer occurring, but we'll save that for the next video. Okay, so remember, the reason it's so slow is because it is not actually that dangerous at all. And the other thing that you have to acknowledge is that the human papillomavirus does not leave the epithelium. It only infects the epithelium. It does not go into the tissue underneath. It does not go into the blood. It is not that dangerous, which is why you do not launch a very powerful immune response against it. Okay, we do not really have a good understanding of what the immune response eventually actually consists of. There's a lot of controversy about whether it's cytotoxic T lymphocytes. So a lot of people think it's cytotoxic T lymphocytes, and then other people are like, cytotoxic T lymphocytes don't actually exist outside of test tubes. So there's a huge amount of controversy about what the immune response that eventually does clear the HPV infection from the squamous stratified epithelium is in this 90% of cases where it does get cleared within 2 years. So we're not going to actually discuss that anymore. What I do want to say is that it doesn't involve antibodies, so no antibodies. So the immune response does not produce antibodies against the HPV virus particles, and the absolutely key thing to put here is you get no future protection. So normally when you have an immune response, it protects you from ever getting that infection ever again. With the immune response to human papillomaviruses, that is not the case. You can have an infection in an area with a certain human papillomavirus, and that can be cleared by the immune response in, let's say, 2 years, and then you can get another infection somewhere else with that same human papillomavirus. You do not get future protection from getting the infection again, so you do not get future protection from the immune response that might eventually clear the human papillomavirus. And that is in contrast, this last line is in contrast to what you get when we vaccinate people against human papillomavirus. You do get antibodies produced and you do get future protection, but we'll come on to that right at the end of the video. So overall, to summarise what I've said about the immune response, it does occur, but it's extremely slow because the virus is not actually acutely deadly at all. It's not dangerous. It doesn't affect any part of the body other than the epithelium. And overall, the statistics are that in 90% of HPV infections, they will be cleared within 2 years. The other 10% will not, and we call those persistent HPV infections. And these are the dangerous ones. When you get a persistent high-risk human papillomavirus infection, that's when it becomes dangerous, and we'll discuss that in the next video. And overall, the immune response that you do eventually launch is not well understood. There's a lot of controversy about what actually happens, but overall we do know it doesn't involve antibodies being produced, and that you don't get future protection. You can get infected again with the exact same human papillomavirus type somewhere else, and you don't have any protection against it, which is in contrast to what you get when we vaccinate people against human papillomavirus. Okay, so I'm going to have a break here. In the next video, we will talk about how having a persistent high-risk human papillomavirus infection of the squamous stratified epithelium of the ectocervix can lead to squamous cell carcinoma of the cervix.